Okay. Well, people come in and get settled. I had the distinct pleasure of introducing my friend and colleague, Dr. Pat Seed, who is a faculty member as well at Duke University. He has MD, PhD from Rochester, is that right? The University of Rochester. And our paths crossed at the University of Michigan. And then he went to WashU in St. Louis and eventually down south to join the faculty at Duke. And Pat's been doing some really just amazing, fascinating, interesting work around the microbiome, which she's going to talk to us about today as our afternoon keynote talk. And the thing that's, I think, particularly interesting is that, you know, after listening to a talk about the microbiome, you start to think, well, gosh, anybody should be able to do one, single organism, one human. How hard could that be when you start listening to the number of organisms that Pat studies and the interaction amongst all of them. So I think we're very lucky to have Pat here to kick us off this afternoon.
Thanks, Alex. All right. Well, thank you very much, Alex. That was quite kind, and thanks for having me. I appreciate it greatly. After you've all had your lunch and we get to talk about the bugs that are going to help you deal with that this afternoon. So it all works beautifully according to plan. So as Alex said, I'm at Duke University. My lab actually studies a variety of molecular events around host microbe interactions, and we actually principally study the microbiome in preterm infants as well as some other areas. So actually I'm going to talk very briefly. You'll see a couple data slides from ours to illustrate a few points, but I'm going to talk a lot about work of my colleagues as well to try and put together a big story for you, to try and understand a little bit about issues about how the microbiome is distributed across the human body, a little bit about how we gain the microbiome and how it develops over time, so a little bit of microbiome childhood development. And then I want to talk about microbiome in disease states. And finally, I'm going to go at the end and talk about a pretty early area of the microbiome where there's an intersection between that and understanding human genotypes and trying to understand whether there is a direct relationship between the type of microbiome we acquire and we maintain in our bodies, as there is with our genetic background. And again, those are early studies, but I wanted to highlight those at this point. So I have no conflicts to disclose, and I'm not going to talk unless you think fecal transplants are medications. I'm not going to talk about off-label about anything. So I always like to point out right off the bat that the major things that shape humanity, in my opinion, are unfortunately war, and we unfortunately get to hear about those a lot right now, and are infectious diseases, okay? These are the things that have really changed human populations over the millennia a time. But I would argue that actually the postulates of Koch and Pasteur, brilliant men, huge contributions that we still refer to, actually make us a little bit narrow-minded and about thinking about our interactions with microbes, where we've really been taught and we think over time about this idea of one microbe causing one disease. And I think as we go through this talk, you'll see that this is only the tip of the iceberg about our interactions with microbes and their ability to cooperate with us in both health and disease states. We in fact live in a world of microbes. We're surrounded with these, and I'll argue before we're even born, and I'll point that along the way, all the way through our lifespan. And this is a very complex group of organisms with very different physiologies that they bring to the table. Furthermore, we think of microbes in test tubes that are floating freely and things. It's actually not the way microbes behave. Microbes actually like societies, communities, and they're actually very cosmopolitan and very socialized. They have cues to work with each other. They build very organized structures to interact with each other that even are things like the Roman aqueducts. They figure out how to filter nutrients through channels to direct it to growing populations and things. So this is much more complicated and sophisticated and evolved than that growth in the test tube or that single organism that causes disease. And of course, as you all know, and you've participated in parts of this just now, microbes are critical for food production. They're incredibly important ecologically outside of our own ecology in soil regeneration, detoxification, remediation, pollutants degradation, oxygen production. Many of you don't realize cyanobacteria are the most abundant kind of organism on the planet, and they are throughout our oceans, and they actually convert CO2 to oxygen. So when you hear your marine biologists talk about the killing the oceans, you're not just worried about the dolphins, okay? It's the cyanobacteria, because that's our source, our major source. It's not just the rainforests. And then, of course, human health. So I want to go through a little bit of terminology, because I'll flip between these a little bit, and a little bit of history about where this concept of the microbiome came from. So actually, Joshua Lederberg, the Nobel Prize winner, was actually who coined the term back in the early 2000s. Joshua won the Nobel Prize for looking at genetic exchange between microbes. But through his career, he realized that we were really being limited by studying this Koch's postulate of one organism, one disease, when we live in a much more diverse community. And he said that really the microbiome term should be used to signify the ecological community of commensal, symbiotic, and pathogenic organisms, okay, that share our body space and really up to that point have been relatively ignored, okay? When I use microbiome, as does much of the field right now, we're really talking about primarily the collective molecular features of a group of organisms, okay? So the genes and then their molecular phenotype, rather than what is really the actual organisms that compose a community. That we will call the microbiota, okay? And the hologenome, or the macroorganism, really is this idea that we are really, truly, our phenotype is a collection of both microbes and human parts. Okay. All right, so some facts and figures, because these are always fun, and if you haven't heard them before, they're pretty fascinating. So we are the home to about 14 log, 10 to the 14th bacteria distributed across the body. And this is at least a log over the number of human cells we have. So when people tell you that you're more microbial than you are human, that's actually true. And this is about 13.3 million different bacterial genes, okay, relative to the 150-fold fewer human genes that we have, okay? And this is about, for the average adult, about 3 pounds of your body weight in your gut is microbial, okay? A little take home for you. And this is about, just to put it on a scheme, we think of genera being like Escherichia or Klebsiella or Staphylococcus. There's about 1,800 of the at the genus level. And then people, this is a sliding number, people think there's somewhere in the range of 15 to maybe 40,000 species in that range. But we keep discovering new ones. So this number keeps going up, where we thought we kind of had a handle on that. And we established the microbiome, and I'm going to talk about this in more detail, after the rupture of amniotic membranes, but I would argue that we are clearly exposed to microbes, or a lot of us are, prior to that birth event, okay? This is something that really, this idea of us being born sterile, I think is in question right now, whether that's the case, all right? And really, the microbiome is something that I'm going to show you is surprisingly stable, but actually undulating within us. It finds a set point, a homeostatic point, and it varies around that. And this is kind of pretty cool data that I'm going to talk about circadian rhythms in the microbiome, okay? So just a little bit of detail. I'm not going to talk a lot technically, but just so that everybody's on the same page about how we examine the microbiome. We don't culture all these organisms. We actually realize that over 90% of the organisms are non-cultivatable in the body, okay, that we're interested in. You just can't do it, okay? So this is all molecular techniques, and we use marker gene studies, no different than you would in human or mammalian studies. So we take a microbial sample, we purify nucleic acid, and classically people use ribosomal RNA, particularly the small subunit, the 16S subunit, as the marker for sequencing. And there are very conserved regions of this with then characteristic SNPs that are dispersed between those conserved regions that can then be used to assign a taxonomic level to it, genus, class, family, et cetera, okay? And we then do bioinformatics and sort this out based on large reference sets of the assignments of those marker genes. And in other cases, we'll actually take all the nucleic acid, shear it, and sequence everything. And you can imagine that's a pretty sticky wicket. There's a lot of gene material coming from a lot of different organisms. And then we sort this out. And in studies that I'm not going to highlight quite as much, of course, there's things like metatranscriptomics where we'll look at RNAs that come from microbes. There's proteomics and metabolomics, and these are all incredibly important parts. They just don't have the same resolution as this. So I'm going to highlight a little bit more the resolution from these types of approaches. So first, let me talk a little bit about the distribution and the different distribution throughout the body. We really are like a rainforest where if you fly over it, it looks like a forest. But when you really get in there, there's a lot of sub-niches. There's a lot of diversity and small ecological niches that are present there. And this is highlighted really nicely in the skin microbiome. This is work from Julie Segre's group at the NIH, where they actually then took a series of these are all healthy individuals, and they sampled serially over these individuals a wide variety of body spots. And you can see these pie charts represent different classes of organisms. Some are at the family level, some are at the phylum level. And you can see that the distribution changes throughout the body. It's not really surprising if you think about it. Some areas are wet, some are very dry, some have higher pHs than others that are lower pH, and some then will have different immunological interactions that are present as well, okay? So a lot of diversity that's even over one person's body. If you look at it on an organ level, more broadly, these are data from the Human Microbiome Project, so these were all healthy individuals. And what you're looking at here is a principal coordinate analysis, and many of you know what this is. For those of you who don't, we'll just think of it very simply that this boils down a lot of very sparse, broad variables into the determinants that are most characteristic of a population. So we can put it in 2D. We can show you all this mass, trillions of sequences down into a little 2D spot. And it shows you the relative similarity of each of these samples to one another. And what's very clear is that your gut microbiota, those organisms in the gut, are very separate from the urogenital, from the oral, from the nasal. It's not to say that there aren't shared members, but generally speaking, they're very different communities in their structure and their composition, okay? But then we can dig down a little bit in one of those communities. So in this case, we're looking at the gut, and you can actually then see across healthy individuals, you can see subtypes. So in this case, we're calling them enterotypes for the enteric tract. And you can see that there are individuals who have a different group here versus here that are characterized by very abundant sort of dominant organisms. So in this case, it's Bacteroides. Down here, it's actually Prevotella, and this is a Ruminococcus group, okay? So we have, within individuals, we will have this separation among individuals of dominant types that are present. And that's true in other places like the vaginal tract and things. We also have to remember that the microbiota has been evolving with us for a long time, okay? And they've done some really neat studies where they've actually done paleo microbiomics, where they actually go in and they actually find ancient remains of humans, and they get gut contents, and they actually ask what's there and how similar it is to modern day. So this is 2 ancient humans that were from caves down in Mexico over 1,000 years ago. And what's really fascinating about this is if I showed you modern individuals, you'd see a similar distribution of organisms. So the distribution's the same. Generally, the types of organisms are there. But when they then did analysis of what are KEGG functional groups to modern groups, so they sequenced everything and asked, how similar functionally might this be predicted to be compared to modern humans, they found that they're significantly different. Now, I can't say that that's a human genetic evolution thing. It could be dietary as well. These were probably primarily a hunter-gatherer population. But it shows that there is a major difference between the modern industrialized world in terms of function, while our composition is the same. So the organisms are probably contributing differently because of difference in speciation. Okay. So I want to take everybody through this idea of the development of the microbiome and our interactions in early life with the microbiome as we develop it. And really, I'm going to start before infancy, because actually the microbiome has an impact on pregnancy that's really interesting data that have come out in the past 2 years. So this is a study by my colleague Ruth Lay, who's at Cornell University. And what she did here was they took a series of pregnant women, they sampled them at different trimesters through their pregnancies. These, I should say, were healthy, full term. They went to full term after they followed them through. And they compared their gut microbiota, the surrogate was stool in this case, in the first trimester, the third trimester, to men and women in this healthy, non-pregnant human microbiome project reference set. And what they saw here was that they saw that here are all the HMP individuals, okay, that are present here. They saw that there was a change in the overall community structure in the first trimester, and then a more dramatic dispersion that happened in the third trimester. So the microbiome was changing in concert with pregnancy as pregnancy progressed. And they show here, this is showing a phylogenetic distance, so a relatedness in the communities more quantitatively. Here's more different and here's more similar. And you can see here the healthy pregnant controls, that there was a difference between the first and the third trimester. They then looked by body mass index, and they showed that overweight women had a similar change in the first to the third trimester, but that this then varied. The obese individuals had less of a dramatic change, and that was even less in gestational diabetes women, okay? And here's the punchline for this, and I'm going to summarize it because of time, and I want to get through some other parts. So they had this model where there was healthy women have a normal microbiota as they enter into pregnancy, and during this time, they start to change their microbiota to one that looks more like an obese microbiota in non-pregnant individuals, okay? And that may be this state of temporary insulin resistance that occurs during pregnancy. This is a natural physiologic state to get more substrates to the fetus, and that this is then a temporarily altered microbiota. So to sort of prove that the microbiota itself, though, this is just association, right? I've just told you what they observed through this. To give a better causation for the microbiota driving a increased body weight phenotype, they took advantage of a technology that we use very frequently, which is that we take gnotobiotic animals. These are born sterile. They have no microbes when they're born. And what they did is they transplanted in microbes from the first trimester or the third trimester. So they went human to lower mammal. And what they found was that those animals that got the first trimester had a normal body habitus over the first 2 to 3 weeks. Those that got the third trimester transplants then had lower insulin sensitization or sensitivity, and they also became fatter. Okay, they gained weight. So the microbiome changes during pregnancy. It changes the insulin resistant state in the pregnant woman, and that is then driving this increased metabolic load to the fetus. Okay. And then after that, you have other early encounters with the microbiota of your mother. So this is a study that we've conducted where we actually looked at the fetal membranes. So these are the membranes from the amniotic membranes from right around the infant. And we looked at these from women who have term labor, preterm labor, or premature rupture of membranes, preterm labor, and then 2 sets where these were actually cesarean sections for term or preterm birth. So these never had rupture of membranes, they never went through labor. And what you can clearly see is that even in those that were from a surgical process of getting these membranes, they are teeming with different kinds of bacteria, okay? So right at the membranes, at that interface of mother to fetus, there are tons of bacteria. And I'm not going to show you this, but a Baylor group recently showed that also the placenta, taken out under surgical conditions during C-section, also has a microbiota that's resident in it during pregnancy, okay? So microbes are making their way to the entire maternal fetal unit at some point in pregnancy, and we don't know exactly when, okay? And this is going to be important when we think about metabolic and inflammatory programming, right? Okay. So in another study we conducted, we were trying to look at the relationship between the mother's microbiota and the early infant's microbiota. And so this is a network diagram. Let me step you through this. So all of these you can think of as species from the mother's stool. All of these are the baby's stool that are in green. Breast milk are in pink. And actually, and I'm going to just a little bit perverse, but this is actually blood, is the mother's blood in red. And what we're able to find is at a very good genetic resolution, we're able to find not only connections between breast milk and the infant stool, that makes sense, the infants are getting maternal breast milk, but we can also find connections between the mother's stool, the blood, her breast milk, and the infant stool in a model that we call enteromammary trafficking. And it's long puzzled me why breast milk has gut bacteria in it. I've known this since I was a pediatric resident, and it bothered me. And we think this is a mechanism that organisms are actually in pregnancy trafficking to the mammary organ and being delivered for reasons we don't understand yet. I could tell you it's conserved across mammals because you can observe the same thing in rats and mice as well, okay? So there's a very early connection, this delivery of microbes that interconnects mothers and infants, okay? So what happens then over the first several years of life? So this is another study from Ruth Lay, and this is highlighting one mother and one infant to make the point for this. This is the first principal component on those principal coordinate analyses I showed you, and it's just showing us again an estimate of divergence, of difference between microbial communities. So here's moms down here, and this is at birth over here. We're looking at age and days on the x-axis. And what you can see is that at birth, the meconium, that first stool, is quite different than the mother's. And as we proceed over the first year of life, we become very similar to then the mothers, and this persists over time. And you can see that this is through breast milk feeding and then actually precedes even the transition to weaning of breast milk, okay? So again, it takes a while, even though there's this interconnection with your mom, it takes a while for the baby, the infant, to start to assume a more adult or mature microbiota, okay? So sort of in summary, what you see is this, is that we start with a very low diverse microbiota. It's principally gram-negative bacteria. These are your Escherichias and your Klebsiellas and your Citrobacters. And it starts to diversify by 1 month of age, where we start to get more bifidobacteria, those probiotics you think of, some anaerobes like Clostridia. And this will continue until we're in our toddler years, when we really start to get much more of a entrenched adult-like microbiota, where we have these classic anaerobes like the Bacteroides, okay? And of course, the human gene content stays stable, relatively speaking. I know it's phenotypically diverse or dynamic, but the microbial gene content starts from about 10 to the 3, 10 to the 4 genes, up to about this 10 to the 6th, almost 10 to the 7th genes in some individuals, okay? All right. So the question that came up over at lunch is, how stable is the microbiome in a healthy individual? And the answer is, it depends on how you're looking at stability. So let's ask the question on a day-to-day basis over time. So a friend and colleague of mine, Lawrence David, did this study that was published in the past year and a half. And what he did is he had multiple individuals, I'm showing 2 of them here, where they sampled every single day, and they actually used an iPhone app to take in all their dietary and and literally is when they defecated and urinated and slept and all these things. And they tracked this over time. Here you're seeing at a phylum level these streaming graphs of the different types of taxa that are present. And here you're seeing the relative change. So when you see green and yellow, things are relatively stable. When they're blue, some group is changing in this. And you can see that these individuals are relatively stable over this period of, here's days, right? So, you know, it's a year, almost a year in both of them, except for a few events. And one was the transition abroad, so diet changed some. So this individual went abroad during this period and then came home, okay? So they got this big ecological catastrophe, and then things changed back. And then subject B got salmonella gastroenteritis. They had a severe salmonella diarrheal disease and had this change. But generally speaking, the message is, it's pretty stable, okay? Now let me shake that story up a little bit. So this was the the thought at this point. But there was then a group at the University of Chicago that has been really interested in diet and the microbiome. This is Eugene Chang's group. And they were looking at mice that were conventionally colonized, they got normal microbes, and mice that were gnotobiotic. So these are those sterile mice I talked about. And they looked at signatures, so these are transcriptional profiles in the liver. And they noticed something really peculiar, that they noticed that the comparison between the two groups, there was dysfunction in the gnotobiotic animals in circadian rhythm signaling. And they thought, why taking away microbes in the gut and on the body of these animals, would that change circadian rhythm? And so they set up a very impressive study where they were able to serially sample the gut microbiota in animals. Whoops, sorry about that. And I'm just showing you part of this. So here's a 24-hour clock day. And what they're able to show is animals that got normal rat chow, they have these organisms that are in blue that undergo this diurnal rhythm. Okay? It's oscillating. It's very impressive when you look through all these data. When they changed the diet, either to high-fat or took obese animals, in many cases, this rhythm flattened out. Okay? And so their model is that the microbiota is, as part of this obesity signal, needs normally to interact with our circadian rhythm and to program these ideas of hunger and satiety, okay, at the hypothalamus, okay? So hopefully all of yours are, in proper rhythm right now, and you guys are all feeling full after lunch, as you should, okay? All right. So let's talk more broadly about what the microbiome does. So the microbiome is involved in a series of different processes: metabolism, immunity, allergy, inflammation, the gut-brain axis. I'm only going to allude to that shortly, and it's been implicated in cancer. And this is both in direct interaction with the host, and there's sort of emerging data about potential epigenetic changes that may be induced by different microbiota. Okay. So, of course, the microbiome will help us harvest nutrients and energy, produce vitamins, metabolize xenobiotics that we're exposed to all the time, provide resistance to tumors and cancers, and help us develop our immune system. So let me talk through some of these features. So, of course, we are innately, and I say that purposely, we're innately programmed to recognize microbes. We have a wide variety of receptors. Many of you study probably polymorphisms in these and susceptibility to disease, okay? And we are programmed both with cellular and intracellular receptors that are important for immunity and tolerance, metabolism, and inflammation. And there are data that suggests that as you have your major colonization events, there's probably some order that we need to press all of these receptor buttons, these toll-like and nod-like receptors that are present. So we can ask the question, well, what happens if you don't have a microbiota when you're born? And the short summary is that you have decreased lymphoid tissue, decreased vascularity of the gut and more distal to that. Mucosal cell turnover is lower, which is part of the cancer model, that you don't get rid of myeloplastic or dysplastic cells, rather. Cytokine production changes and digestive enzymes change. And you have increased caloric requirements. You just can't harvest nutrients, so you need more. And there's an increased susceptibility to infections. And this, I think this panel nicely highlights some of the changes in immune function. So this is staining for T cells, for T helper cells, CD4 cells in the spleen and the intestine. And you can see that in a germ-free animal, they have much less of this sort of pink and brown stain that's present in both sets of tissues. And similarly, there's less secretory IgA present in the germ-free animals than the normally colonized animals. Okay? So to illustrate this further, here's an experiment that shows you what happens if you have inappropriate or delayed colonization. So in this experiment, they took normal animals and they colonized them. They colonized gnotobiotic or germ-free animals at different times. One early at 1 week and one later at 3 weeks. And what they were able to show was that now at 2 months of age, so well past these events, okay, they were able to show persistent changes in what are mesenteric lymph node responses to lipopolysaccharide, or the gram-negative toxin, and IL-12, TNF-alpha, interferon gamma, and IL-6. So this delayed colonization produces persistent effects. These are in a mouse's lifetime. This was getting them through adolescence, early adulthood, okay, and sexual maturity. So these are really profound events early on in terms of programming, inflammation, and immunity. Similarly, if you deny an animal colonization, you see an upregulation in these receptors, sort of like the host is seeking those signals that were missing, and those are present throughout the intestinal tract. And if you actually remove these switches, so in this case they're taking out TLR5, which senses the flagellin, that waggly tail that some bacteria make. If you take that out, these animals will actually have an obesity phenotype. They actually have increased food intake, increased calorie use, and increased insulin and glucose. They actually have an absent satiety response. Colleagues of mine who study these, they say the animals are just voracious eaters. You literally have to like pull them away from food because they just want to eat. That's just from taking away this one receptor in the gut. This is gut specific, TLR5, okay? And when you transplant, again, the microbiota, the feces from this knockout mouse into a germ-free animal that's normal, you now get an obese phenotype, okay? So this knockout produces a microbiota that in itself is obesogenic, okay? All right. So what I've been talking about now is abnormal communities, the change from normal development and that stableness that I told you into communities that have shifted, they've changed, they have these other phenotypes like conferring obesity. And this is called dysbiosis, or the imbalance of the structure of the commensal microbiota, the normal microbiota. And this has been described for a variety of processes: antibiotic-associated diarrhea, inflammatory bowel disease, irritable bowel disease, necrotizing enterocolitis, obesity, eczema, type 1 diabetes, autism, systemic lupus erythematosus. There's a dysbiosis with a variety of different important human diseases. Ooh, that wasn't good. There we go. Okay, so the classic study on dysbiosis is the one of obesity. And I wanted to start with this one from Jeff Gordon's group, where they looked at individuals, these are humans, who were weeks on diet, they were obese or they were lean, and they looked as they dieted and they saw that there was this increased ratio of firmicutes to bacteroides early on, and as they dieted, they assumed a more lean-like ratio on that diet, okay? And what they did is they then showed that the microbiota in mice that are leptin deficient, that these mice now had increased acetate production, increased butyrate production. These are all substrates, right? Metabolic substrates. So they're actually making more energy for the host to drive obesity. And this same obese phenotype in these mice, or obese microbiota, extracts more energy. It takes more kilocalories out of the feces from the diet, and there's a subsequent increase in body fat. With inflammatory bowel disease, there are major rearrangements in the microbiota. Here's a normal set and an IBD set, and you can see this bloom of the gram-negative proteobacteria primarily. And you can see this is now looking at biopsy samples throughout the intestinal tract. And you can see that in the normal versus this dysbiotic, this altered one, there's huge shifts that really affect the entire gut in inflammatory bowel disease. Okay? This happens in processes like bacterial vaginosis, where if we look at the similarity between different groups, in this case no vaginosis versus with vaginosis, you can see that with vaginosis there's increased diversity in these microbiota versus those who don't have BV, and there's a blossom of unique organisms that are present during that process. Okay, so that's all examples of dysbiosis, but let's talk about really common things that we do to produce early life dysbiosis. So let me tie this back to the infant story. So one of the common things we do is we give antibiotics to young children and into livestock We flood our foodstuffs with exposure to antibiotics. So a group led by Marty Blazer at NYU has been interested in early pulsatiles. So these are antibiotics that look like the way we would normally get them, whether we're administering to infants or through food, and looking at the results on not only the microbiota, but phenotypes, and in this case, obesity, which is a big area of interest for him. So mice were administered one of 2 antibiotics in the first 2 weeks of life in this pulsatile fashion, and control animals didn't receive the periodic animals. The 2 regimens they received was one amoxicillin, the classic ear infection drug, and the other one's something called tylosin, and it's the leading drug used in veterinary practice to make fat animals, okay, is basically what it's used for. And what they showed was those animals that had early exposure, this is relative to the controls, had increased body mass, not only shortly after they received that, but out 15 to 24 weeks. These are old in mice time, these are old mice. They had persistent increases in this body habitus, this fat body habitus, okay? And particularly with the tylosin group, okay, which is pretty concerning. You can see that if we look at, these are different types of organisms across class and family, and you can see here's the control distribution that's present. Here's the antibiotic treated one with amoxicillin. Here's tylosin, which really is a decimation of this microbiota, and here's the mixture. So these are producing pretty profound dysbiosis. Okay? And what they showed is now these adult animals, so these ones that were exposed to antibiotics early on, they showed that they have very different transcriptional profiles in the liver even after they're adult animals. So this is RNA-seq transcriptomics of the liver in animals that got tylosin versus amoxicillin compared to the controls. And there's an increase in a variety of hepatic functions, synthesis of fatty acid, eicosanoids, so inflammatory pathways, et cetera. And you can go down the list, okay? And that's particularly remarkable for tylosin. So establishing or disrupting this microbiome very early on is critical, at least if these animals represent the human state early on. And in fact, there are epidemiologic data, I should add, that early antibiotic exposure in human infants actually has an increased propensity for obesity. So there is an epidemiologic background to suspect that there is a connection here in this model, okay? So I want to sort of end this last part with some fundamental questions about, does host genotype then make it more or less likely for us to get different microbiome states, different types of microbiomes, okay? And so I'm going to talk about a couple twin-twin studies initially, and then I'll talk about a few SNP association studies as well. So here's a study from 2009 from Peter Turnbaugh when he was with Jeff Gordon at Washington University as a grad student postdoc. And here we're looking at one of these graphs where we're looking at the phylogenetic distance, so the similarity of communities. They're either more different up at the top of the y-axis or more similar at the bottom. And here are the different relationships we're looking at. Here's the relationship. I'm going to start from over here from your right, where the relationship, of course, of self to self is very high when you serialist sample that. Monozygotic and dizygotic twins, so twin-twin relationships, were not significantly different here, but those were more similar to each other, their fecal microbiota, than were twin-twin to mother, okay, relationships. And then, of course, unrelated are the least related to these twins, okay, all right? So that was interesting, but it was a small group of twins, okay, about 100. So this is now a study from Ruth Lay's group at Cornell, where they collaborated to look at twin-twin cohorts in Northern Europe. So this is Northern Europe, to keep that in mind alone. And they looked at the relationship then between monozygotics, dizygotics, and unrelated. And they actually found that, in fact, monozygotics are most closely related to one another versus dizygotics versus unrelated. And they found that for very specific organisms as well. So it seems that there is more of a genetic relationship or a component to you having certain organisms like Ruminococcus, Lactobacillus, and Bacteroideaceae, okay? All right. Here's a haplogroup bacterial family association study. So this was, these were subjects that were taken from the Human Microbiome Project, so healthy individuals, all on American diets. There was nobody in this who had vegetarian diets or unusual diets with this. And they took 89 subjects, and they subgrouped it because they wanted multiple data sets that they needed available to them. They actually used, in this case, mitochondrial DNA sequences for the haplogrouping, okay? And what they observed was that the microbiome samples in a variety of different sites, from the vagina to the stool to nares to the dorsum of the tongue, had different profiles based on the haplogroup, okay, that was present here. They went on to show that there were very specific organisms that were enriched, significantly increased, associated with different haplogroups. In this case, this is the gut that they're looking at here. They then clustered, they grouped together the types of microbiota. So they clustered this whole thing and said, okay, which microbiota kind of go together? You can see the clusters that are up here that are formed. And they asked which subgroups or clusters then are most associated with the haplogroups. And it seemed that different microbial compositions, groupings, were more associated with different haplogroups in the study. And that was in the vaginal studies and in the fecal studies as well. Okay. And last, when they looked at a series of SNPs across that, they found very specific SNP associations with enrichments for certain organisms in both the feces and posterior fornix. So this study is good in the sense that it represents individuals across the US, not one center or regional, but it's relatively small. So we all take this with a grain of salt, but it's very provocative, this idea that there are genotype, haplotype microbiota associations. And last, let me talk about a inflammatory bowel disease association study where they were assimilating microbiome, whole genome, SNP analysis, and environmental factors, so clinical data, to look at associations of inflammatory bowel disease susceptibility loci in the microbiota. And so here they're looking at a number of known risk SNPs in IL-12 and a series of other ones that are present here. And they're looking at gene dosage, and they show that having 2 copies, you actually have an increased risk for some of these organisms based on gene copy that's present here. And they focus down on, of all of these, on NOD2, the intracellular receptor for lipopolysaccharide. And there's a number of known risk alleles for this. And they looked at those, and they showed that there is a dosage effect for Enterobacteriaceae. And Enterobacteriaceae are things like E. coli that have been clearly implicated in both the onset of inflammatory bowel disease and also the relapse of inflammatory bowel disease. Okay? And when they put this together, they showed a relative risk based on the clinical profiles, antibiotic or biologic exposures, other drug exposures, biopsy phenotypes, and host genetics, for there being a connection or a likelihood that there would be different microbiota present in the guts of these individuals. And I present this because I think this is really early and needs to be replicated and needs to be done in larger study sets. But I think that these are going to be the kinds of predictive signatures that we might see down the line, where we're incorporating gut microbiome and human genetic data with classic clinical risk factors for both disease onset as well as disease either response to therapy or relapse of disease. Okay. And last, I'm going to finish with 3 slides, despite my 0. And this is because I want to leave you with, well, what do we do with all of this? Because we have these changes, they seem to be associated with disease. How can we manipulate the microbiome? And right now, the state of the art is a sledgehammer. But I want to highlight the sledgehammer and where we're going to go from there to show how profound manipulation of the microbiota is. So many of you are familiar with Clostridium difficile diarrhea. You get antibiotics, you get this dysbiosis I described, and this organism, a toxin producer, grows out in your gut and causes an inflammatory colitis that's present. So this group in the Netherlands did a randomized controlled trial where they took individuals who had refractory recurrent C. difficile, and they gave them either vancomycin orally, which is a classic clinical treatment for this, or they gave them infusion of stool. And what they showed was that infusion of stool from a healthy donor was far superior to the classic antibiotic treatment that they gave. In fact, they stopped this study early because the differences were so profound. They felt it wasn't ethical to continue to not offer the transplant to the vancomycin group, okay? So profound alteration of people who, these people all went in with about a year of chronic C. difficile into the study. They get one dose, and the vast majority of them are cured, gone, okay? Here's a study where I told you about the obesity and insulin resistance. Here's a human trial where they took insulin-resistant individuals. They gave them allogeneic or autologous transplants. So they either gave them their own stool back or they gave them a healthy, lean donor stool. And they measured insulin resistance. And look at this. Before and after the fecal transplant, there was an improvement in insulin sensitivity after this. Small study, but how provocative is that? Okay? No drugs. We're going to give you different microbiota from lean, insulin-sensitive individuals. And last, I'm going to go to animals, but this is, I think, very provocative, which is the connection of gut-brain. And this is a study where they took animals that were naturally prone to anxiety, and these animals, when they're stressed, will have very high cortisol levels. And they found when they gave this probiotic, this lactobacillus, that it actually would reduce stress under those stress conditions. Just the administration of a probiotic did this. And there's actually a mechanism for this, which is that this enzyme has an ability to convert glutamine to GABA aminobutyric acid, okay? It can convert it and make the neurotransmitter that actually will decrease anxiety functions, okay? All right. Oh, and I should highlight with this, if you guys look this up, that if you sever the vagus nerve, that actually ablates this effect. So the effect is gut through vagus nerve, okay? All right, so in summary, our microbiome is 150 times the genetic content of the human microbiome, and it's really specialized throughout our body. The microbiome is critical for all these broad physiologic effects that I described, and dysbiosis may unmask, initiate, or collaborate in diseases. And shifts may be associated with normal processes that we just haven't studied enough, like pregnancy. Early human genetic microbiome studies suggest that there may be links fundamentally between the human genotype and the type of microbiome communities that we associate with. And I think this needs more study. And so I think the future is going to be finding combined predictors of disease that are human and microbial. Understanding mechanisms remains a major issue in this field. Investigation of compatibility types or discompatibility types, especially as we think about transplantation and modification. And then this idea of personalized human microbial genotype matching for microbiome transplants and probiotics. And last, I think very relevant to enzyme deficiencies and things, is going to be this idea of engineering. How do we use microbes to bioremediate or give back things that we're missing fundamentally? And I think this is a really promising area that's very early in its infancy. So I just want to thank a lot of funding sources and my collaborators who helped me with my own work. Thank you very much.
Talk like that and not give a chance for people to ask questions. So
that was great. I had a quick question. I've read several studies that link the use of PPIs, proton pump inhibitors, with necrotizing enterocolitis, and I'm wondering if anyone has studied if changes in the microbiome are actually responsible for this relationship.
So that's a great question. So everybody heard that. It's the proton pump inhibitors decrease acid in the gut, and infants will get this bowel necrosing disease that's common between preterm infants. And the mechanism is thought to be decreased acidification and killing of organisms as they're swallowed that are pathogens. So that's clearly been linked epidemiologically. We've actually studied that some, and actually have work that's about probably halfway through that actually shows that there is in fact a dysbiosis in those infants. And there's an enrichment of gram-negative organisms which have also been linked previously, and more recently people have come back to it with necrotizing enterocolitis. So I think there is a story there.
Thank you.
When you go to make these changes in the microbiome, and you go to change the populations, do the microbes fight back? Do they try to stay in at the population level they are, or do they just sort of go with the flow, whatever you put in there?
Yeah, excuse the pun.
Yeah.
Sorry, I had to get that in there. So the story's very interesting, but one that I can't give a straight answer to. So it's interesting, if you actually give animals or humans these whole transplants, you take a whole community and you put it in, there are studies I didn't show you where those people's microbiota will look much more like the donor than their original one. So there is a change. If you look at most of the probiotic studies that have been done, where they've tried to culture the probiotic or look at it molecularly, most of them don't take. There's a small number of individuals that actually that community allows sort of the new guy in the neighborhood. Most of the probiotic effects are transient. They're things passerbys that maybe do something metabolically or inflammatory. And that, I think, is a $100,000 question. Maybe we're in a new era, $10 million question. Because I think people, for these ideas of engineering or supplementation or complementation of human defects, we need to know that. How do you establish it so somebody doesn't have to take it like a drug, because maybe it's not better than a drug if you have to take it every day, right? And so people are trying to work on those rules of the road, but there aren't signs on the highway yet.
Thank you. It was an excellent presentation. I have a practical question. We tried to collect the fecal samples, and we had such a difficult time. I thought it was going to be very easy to collect the fecal samples with the tools we had. Is there any incentive you have given to clients to collect? No,
it's compensation for their time that they spend with us.
No, so are there special,
So I'll tell you right now, babies are great.
Yeah. But these are adults.
I'm joking. That was a joke.
Yeah.
So it's actually, our experience has been very population dependent. Those who have serious disease are willing donors. I think we all have that experience with clinical research, that they are much more likely to participate, and consent rates are higher and things. Normal, healthy individuals, there's a yuck factor. We can't always figure it out. People will let us draw blood, which is usually a big clinical barrier, more than collecting stool. We've actually had women who are more likely to let us get vaginal swabs than they are to collect their stool. So we have used, the answer is yes, we have used a monetary compensation for the time and effort that they've given us.
And for the dose for fecal transplant, is it dose dependent, weight dependent?
No one knows that, and my guess is that it's actually profoundly, there's a profound window of dose, and you can actually use a very small amount. We've done some transplantations in kids, one with metabolic disease who had recurrent C. difficile, and we gave actually pretty small amounts because of donor circumstances to that, and the child was completely cured by that in that circumstance. So it's pretty interesting, but nobody's sort of said this massive stool or things like this.
Just two very quick. I thought Tim got up here because he was going to tell everybody that we have hand sanitizer at the GGC booth, just in case. But secondly, lots of the things that you were hitting on relate to autism or potential areas for autism, and lots of us have interest in that. And I know most of the new literature related to microbiome and autism, if you search it, has really just been published in the last year or two. So I was wondering if you might give us a tidbit of what we should be watching for.
Yeah, so I think there are going to be some larger multi-center studies that are going to look at this. There are going to be a lot of animal models as well as you take, so some of the scaffolding protein defects at neurotransmitter synapses that will look at some of these effects right now. There's been actually talk about, there are a lot of parent advocate groups that would like to do some transplantation studies based on what's been done. I think it's way too early for that. I actually think that right now, the Microbiome Association studies are pretty mixed. There are associations with a dysbiosis, but there are also some studies that are pretty unimpressive that there's not a big mixture. So I think we have more just association to look for before then we start to ask, are there keystone organisms? Or you probably saw from Caltech, Sarcus masmani, and it's an impressive study in animals, right? That they found a small molecule that seems to be made by the microbiota, that if you infuse it in animals, they get autism-like features. I think we have yet to validate what really is maybe a microbiome phenotype of humans, particularly at different ages when they're developing their microbiota. It's changing dynamically before we sort of leap there.
Yeah, no, I think that's probably good advice. The thing that we've talked about back in Greenwood quite a bit is the notion of something happening early on and then disappearing. So whatever the etiology is sort of a ghost, so to speak. And this would be a perfect explanation if there was some sort of predisposing sort of setup for it early on that sort of changed over one's lifetime.
The mechanism is not clear, but there are clearly, I gave you a number of stories of early exposure, normal microbiota after that, and then this persistent phenotype. So I think you're very wise to think about a window of opportunity or disopportunity. Yeah.
And
Okay.
I'm going to have to
Yep.
take the chair's prerogative and ask you to stop, but I'm sure that Dr. C would be more than happy